Thank you. Good morning, everyone. I wanted to speak about focus ultrasound treatment of cerebral cavernous malformation. It's my very initial results. And a brief introduction: cavernous malformation, a low-flow vascular lesion composed of thin and dilated vascular channels, and CMs causing neurological problems in brain or spinal cord. And the incidence of cavernous malformation is estimated at 1 in 100 or 200 people. And a minimum of 30% of people with cavernous malformation will develop symptoms. And here is the worrying part: the most in their 20s and 30s. And at now, we have just a surgery option for treatment. And because of this, we decided to study new therapies and develop new treatments for this disease. And for this experiment, we decided to use CCM mouse model, and we know the CCMs arise from endothelial cells loss of CRIT1, CCM2, or PD-CD10, and we have established inducible endothelial cell-specific CRIT1 knockout, and cavernomas are induced at postnatal days P1-5 with tamoxifen. Really, I inject 250 micrograms of tamoxifen at P1, P3, and P5 intragastrically. And at P6, these mouse start to develop, present some venous dilatation into the cerebellum, and at P11, they have the major lesion into the cerebellum. And in this picture, you can see the black arrow showing the CCMs into the cerebellum. And after this, we decided to do our first experiment, and we got a normal black 6 mouse, inject 5-ALA. After 4 hours, we sacrificed this mouse, perfused with PBS, sliced the brain, and brought this slice to 2-photon microscopy and did this imaging. And here was an amazing result, because the endothelial cells picked up our 5-ALA. And in fact, if 5-ALA can mark blood vessels, it means 5-ALA can target all pathologies with blood vessels, such as tumors and vascular malformations. And after this, we decided to get our CRIT1 knockout mouse P15, and again did the same protocol. And here, with white arrow, we can see 5-ALA into the cavernous malformation, and again label other blood vessels. And with this amazing result, we will do our future experiments. We'll give again 5-ALA to our CRIT1 knockout, and treat this CCM mouse with MRI-guided focus ultrasound. And what we had expected is, if we found some source that say light and ultrasound can convert the 5-ALA to cytotoxic products, and we expected that cytotoxic product can blast the cell of the tumor. And it's our initial results, and in future we will do more experiments and more results. Thank you.
Thank you very much.